Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Integral Molecular, I'd like to welcome you to new approaches for monoclonal antibody discovery against GPCRs, ion channels, and transporters. My name is Elizabeth Lamb, and I'll be your host and moderator for today's event. I'd like to introduce our presenters for today. First is Dr. Ross Chambers, PhD, Director of Antibody Discovery at Integral Molecular. And our second presenter is Dr. Joseph Rucker, PhD, Vice President of Research and Development for Integral Molecular. And with that, I will pass the presenter ball over to you. Welcome, gentlemen.
Thank you, Elizabeth. Thank you for attending, everyone. Okay, so today's talk, we've got roughly a 45-minute talk here split between myself and Dr. Rucker. I'll be going first. So I'm going to cover mAb discovery, and Joe Rucker will be covering mAb characterization against membrane proteins. Okay, so first, I want to tell you a little bit about who we are. Our company, Integral Molecular, is a biotechnology company based in Philadelphia, and our expertise is focused on membrane proteins and antibodies. We've been in business 15 years, and this has been our sole focus. Over those 15 years, we have been trusted by many companies who have used our services and products. We've done a lot of work with many biotech companies and pharmaceutical companies, as well as many collaborations with academic partners, which have resulted in papers in leading journals such as Cell, Science, and Nature. Okay, so that's a little about who we are. So really the topic of today's webinar is isolating mAbs and characterizing them to membrane proteins. Over the last 15 years while we've been in business, we have assembled technologies that addresses the unique challenges that membrane proteins have. And this has now enabled us in the last few years to pull through a system that takes you from target all the way through to high-performance antibodies. And we have assembled technologies to help us get through all these steps, from generating antibodies in animals to isolating the mAbs to engineering them and to characterizing those antibodies. In the first part of the talk, I will be covering these first two sections here on making antigen, immunizing animals, and isolating those mAbs. We won't be covering mAb engineering. We do a lot of mAb engineering here, humanization and affinity maturation. We won't be covering that today. And then Dr. Joe Rucker will be covering mAb profiling, or really mAb characterization, with some unique technologies that we've developed for those applications. Okay, so first up, let's talk about why this is so difficult. So membrane proteins are the most difficult targets to make antibodies against. And the reason is they have a whole bag full of problems that go with them. Listed here on the slide is the top 3 problems that often people struggle with. Number 1 is poor surface expression. Many membrane proteins just don't get to the surface at high levels, which means that it causes problems in immunization, in the assays, in sensitivity, and in isolating the mAbs. And so what we have done here is we've become experts in antigen engineering that we use and deploy to solve poor surface expression problems. The second problem is the epitopes that we're trying to target here are very complex. They are shown in this GPCR structure in red. You can see the extracellular epitopes are often discontinuous from multiple segments. They're small, they're post-translationally modified, which makes them very difficult to hit antibodies to in their native conformation. Because they're embedded in the membrane, when you take the membrane away, if you try to purify these, they fall apart. So what we've done to address that is we employ DNA immunization and lipoparticle immunization to deliver native epitopes to the immune system. The third big problem is that many of the targets that we work with, GPCRs, ion channels, are often highly conserved. This is just illustrated here with a family of GPCRs, and you can see that over half of GPCRs are very highly conserved between humans and mice. So to solve those kinds of problems, we rely on immunizing into other species that are more divergent, such as chickens. So I'm going to walk you through each of these pieces here in the next few slides. Okay, so first up is antigen engineering, a very important step to address. So often, as anyone's familiar with membrane proteins, there's lots of problems with expressing the protein. It can be range from simple things like the codons or things like that that allow them to express, or to trafficking motifs within them, or the protein is intrinsically unstable. And so you have to address these problems in order to get a protein that can be used effectively in any mAb isolation process. In the next slide, I just want to show you an example of antigen engineering, the things that we do to deal with these multiple problems. And there's really no one problem that applies to all proteins. You really have to try addressing a range of problems to getting your target to work. So in these 4 examples here that illustrate some of the issues that you have to deal with, is the first one is a common one is the protein is toxic to the cells when you overexpress them, and so you have to target the motifs to make them inactive but not denature them, to make them non-toxic and express. So in this case here, the wild type was killing the cells, and the engineered version allowed them to express. Another problem is often the protein won't even express. You can look on a Western blot and you won't see anything there, so you have to engineer it to get the levels up. Or in cases of stability, what we employ is high throughput mutagenesis to find mutations that increase stability or improve surface trafficking. I really want to emphasize that you really need to address this step early on, because any problem you skip over here will impact you downstream at the more expensive mAb isolation steps. Okay, so assuming you now have an antigen that's now expressed at high levels, you now want to deliver it to the immune system to get the native epitopes recognized. And the technology that we have relied on a lot is DNA immunization. That's a lot of my background and expertise. So DNA immunization is really an ideal way to present native epitopes of membrane proteins to the immune system. DNA immunization is the delivery of an expression plasmid into an animal. The protein is expressed in vivo. There's no handling of the protein. It's in its membrane, and it's fully native as the immune system sees it. And this is key to getting the native epitope recognition. We have extensive experience with DNA immunization, and we've done many projects with it. Now, one limitation that often people experience with using DNA immunization that I want to address here is getting high titer responses and getting high success rates. The problem with DNA immunization in its plain vanilla version is that you'll DNA immunize an animal and get an antigen-specific B-cell response going. But because DNA immunization generates typically picogram or very low levels of antigen, the immune system usually tilts down to memory B-cell responses, which results in no antibody titer or low antibody titer being produced, and very few plasma cells that produce antibody. So an important modification to this method is to add in a protein boost. The protein boost, even small amounts, nanogram amounts, are sufficient to trigger and switch the memory B cells into plasma cells and significantly improve both the titer and the success rate of immune responses. That's illustrated in this next slide here. In this slide here, I just want to describe how we do this then. So how we immunize is we'll first prime the animals with DNA. This is a pure antigen. We get a single response to the target. And then we'll boost them with a product that we call lipoparticles. These are high concentration particles containing the membrane proteins in their native structure. Both of these are delivered using proprietary adjuvants and special protocols. The combinations of these two together results in very high robust responses to native epitopes. So this is an example shown here. So I want to show 2 examples that show the different kinds of responses you typically see. In example 1, you'll hit them with DNA, and in some cases you'll see that there is a titer being generated after hitting them with multiple times with DNA. However, if you give them a single boost with lipoparticles, you'll see that the titer will increase. However, in many examples, you'll hit them with DNA and you'll see really no response after even multiple DNA immunizations. But then with a single lipoparticle boost, you'll see a massive increase in the titer as those memory cells switch over to producing antibody. So that's really a key point in the success rate of getting good responses. Now, I want to say a word about what you immunize into. So we can do all the usual kinds of animals, as many people do, but we do employ the use of birds in our immunization collection of animals. So birds are valuable here because of their evolutionary distance. So as I mentioned before, many of the targets that we work with are highly conserved. This is shown here in this list of 25 targets that we've worked on, and you can see here that when you compare the sequence to mouse most of these are more than 85% identical between human and mouse, and greater than 85% usually results in immune tolerance and a weak immune response. However, if you immunize into chickens, the evolutionary distance means that most of these are not highly conserved, and you get much better responses. So you typically get responses to highly conserved epitopes, more diverse epitopes, and often antibodies that can see across mouse and human proteins. Chickens also, I should mention, are much easier to humanize than mouse antibodies. Here's an example of our success rate. We've done many different targets, GPCRs, ion channels across all kinds of classes, and we see a really broad and high success rate of getting titer to a range of different proteins. Okay, so we've got some titer. Now we want to isolate the mAbs. Now, one technique that we use a lot is phage display, and many people have used phage display. But really, a key thing here for effectively using phage display to isolate mAbs to membrane proteins is what you pan on. And again, I want to mention the lipoparticles. These have really been key to our success here. Lipoparticles are a high concentrated source of the membrane protein. They display them in the native format, and they're very stable and easy to use. And we have employed this in all of our mAb isolation campaigns to pull out mAbs, and it's been very, very successful. This is illustrated here in a handful of examples I'm just showing you here across many different targets. Typically, we will do a few rounds of panning. We'll then pick a few hundred clones, screen them by flow cytometry, and we'll typically get many dozens of hits from the screening. And the hits often translate into high-performance antibodies. And an additional technology that we have developed recently is another way of isolating mAbs, which is B-cell cloning. And the technology we're using is a microfluidic-based technology where individual primary cells from the animal are encapsulated in these small droplets, as illustrated on the screen here, on this microfluidic chip. The small droplet means that high concentrations of antibodies can be generated in a very short time. What this means is that primary cells can be put in with an assay and read in a few hours while they're still alive. We use microfluidics to sort those droplets and read the assays because it makes the screening very fast. We can screen 200,000 assays every hour, which means we can get through millions of B cells in a day. Because it's B-cell cloning, we get the natural heavy light chain pairing of those antibodies. And it's non-competitive like phages, so we get a more diverse collection of antibodies. So the technology looks like this. So on day one, you will have the animal material, the B cells isolated from the animal. The cells will then be put into these small droplets with an immunoassay, incubated for a short time, and then run through a microfluidic chip to sort out the hits. The hits are then put into single-cell PCR, the genes isolated, expressed, and characterized. So you can get hits within a single day and know if you're on the right track. The next slide is an example of this that we've done here. This is an example with the GPCR CXCR5. In this top panel is showing the assay, the machine run on a negative control immunoassay to get you a sense of how many false positives are coming up. So with these deep screens, of course, the problem is false positives. You want to minimize that as much as possible. So you can see here with the B cells isolated from the animal, 200,000 droplets were screened, and there were really no significant false positives coming through in the screen. However, when you put in the actual CXCR5 assay, you can see a significant shift in the hits and of CXCR5 reactive antibodies coming through. And this is a 500,000 B cells screened here, which is a small fraction of the cells from an animal. And you can screen much deeper. You can keep running for days and days and days. Cells are stored frozen. Okay, so that covers the technology part. I just want to finish up this segment of the webinar with a few case studies to get a sense of what kind of antibodies we deliver. The first target is a glucose transporter. These are very difficult targets in general. We've got 12 transmembrane domains. They've got small extracellular regions. And I want to show you in the slide here, it illustrates the challenges we're up against in that we're trying to target these very small extracellular regions that are poking up above the membrane. The value, again, of using DNA and lipoparticles for immunization is that most of the protein is shielded. The immune system doesn't see all this embedded material in the immunization process. So that means that the immune system is focused on the relevant epitopes, which translates into much more relevant hits, more successful hits, and more functional hits. So we did a campaign with GLUT4, the glucose transporter, immunized into animals, got a good immune titer, made a phage library, in this case, screened the phage, got dozens and dozens of hits. We sequenced those hits. You can see here that you get quite a diverse response. This is families from the heavy chain of the antibody genes. Typically in phage, you'll see one large family and then many, many, many small families. These antibodies, importantly, react in flow cytometry. They were of good affinity, nanomolar affinity. And then critically is, do these antibodies actually work on the real cell? This is a key step. So here we're testing these antibodies on endogenous GLUT4 expression in fat cells. You can see in the small inset, this is cells untreated with insulin. And then when cells are treated with insulin, the GLUT4 is upregulated to the surface. And you can now see that when there's a ring of GLUT4 being detected on the surface of these cells. So these antibodies are able to detect endogenous levels of antibody in a native system. I mentioned before about the value of immunizing into chickens, and this is illustrated here in the ability to isolate antibodies that can detect both human and mouse forms of the protein. So we took 3 of our hits here and showed that indeed that all 3 of these hits were also able to recognize the mouse version of the GLUT4. So this is very helpful for drug development, that you can use the same antibody for animal models as well as the clinical. The second example is an ion channel, P2X3, that is highly conserved. Again, we got a very good immune response in chickens. The antibodies were actually isolated in collaboration with Crystal Bioscience. We showed that these antibodies were indeed conformational, they had good affinity, and then importantly, we're able to find functional antibodies, things that can shut off the ion channel. This is a target for pain. Here in this in vitro assay, you can see it's inhibiting the function, shown in red. And in ex vivo rat nerve cells, you can see that this antibody is inhibiting P2X3 in that. The final case studies is some partner studies where partner had some targets that had failed in their internal efforts, and they gave them to us to have a shot at. In these 2 examples, we were able to rescue these projects. So here in the first example, the problem really was poor surface expression. As I emphasized before, you really need to address this. After engineering, we were able to get an optimized high-expressing construct, and this then resulted in a large collection of mAbs that were able to bind to the real target. In the second example, there was a bag of problems. This was toxic. It was very highly conserved, 99% identical, even to chickens, and the expression was not that great either. However, with our proprietary adjuvants and our immunization system, we were able to induce a very small immune response, and this did eventually result, after much panning, a small number of monoclonal antibodies to this target. Okay, so I want to summarize what I've just talked to you about here. The 3 really main points I want to, as takeaways here, is that one is that it's critical to get high surface levels of expression for any successful mAb program for a membrane protein. It's really important not to skip over this step. Second is that, as I've shown, that DNA priming and like particle boosting is very effective at getting titers against even very difficult native epitopes on membrane proteins. And then third is that with phage panning and with B-cell cloning, like particles have been really key for enabling us to select mAbs against these membrane proteins. In the next part of the talk, I want to hand it over to Dr. Joe Rucker. He's going to cover then this downstream step of characterizing antibodies in terms of safety and epitope mapping.
Great. Thank you, Ross. So, what Ross presented was how, first of all, thanks everyone for joining us for the webinar today. What Ross just showed you is how integral molecular approaches the discovery of antibodies against difficult membrane protein targets. But as I think probably many of you know, is that once you have the antibodies in hand, there are still many challenges involved in characterizing the antibodies as they move through the development process. So what I want to do today is to discuss sort of 2 of these challenges and the approaches that we take for working with these challenges at Integral Molecular. I'm first going to talk about epitope mapping, and how our shotgun mutagenesis platform can tell you where your antibody binds on your target, which of course contributes to a deeper understanding of what your antibody does, and in particular can help you generate novel intellectual property. I will then also talk about our membrane proteome array, which is a new platform that we have here at Integral Molecular, which can really give you an overview, a broad overview of the specificity of the antibody. What are the possible targets on the cell surface that your antibody can bind? And I want to talk about how this can really help you identify safety concerns early on in the development process. So let's talk about antibody epitope mapping. And I think what we have to really start with is, what is an antibody epitope? So on the left side, I've given a schematic of how an antibody binds to an antigen. So an antibody epitope, most simply, is the region of the antigen that is contacted by the antibody. This region is defined by specific amino acid residues on the antigen, that would be the epitope, that contact residues on the antibody, which are known as the paratope. Although many of these residues are in this interface, these interfaces can actually be rather large and rather complicated. Only some of these residues contribute to the strength of the binding interaction, to the overall affinity of the antibody. And we refer to these residues as either energetic hotspots or as critical residues. Now, on the right side, I want to talk about a key way of differentiating antibody epitopes, and that's the difference between linear epitopes and conformational epitopes. A linear epitope is an epitope where the residues are sequential in the sequence of the protein. These are the type of epitopes that you would expect from peptide immunizations and that you would use for antibodies that you were going to do something like Western blotting with. But much more interesting, and in many ways much more important, are conformational epitopes, and that's shown on the right. These are epitopes where the antibody is binding to residues that are discontinuous on the sequence and are formed by an area that's formed by the three-dimensional structure, both tertiary and quaternary, of the protein. And you can see on the snake diagram below that these residues are on very distinct parts of the sequence, but in the three-dimensional structure shown above, come together to form a contact region, binding region. So why do you care about epitope mapping? What good is an epitope for you? Well, epitope mapping of antibodies and defining where your antibody binds can significantly improve the intellectual property and also regulatory approvals of the antibody as it moves through the development process. And this is enabled by the fact that epitope mapping enables you to differentiate candidate molecules in a very well-defined structural way, and also enable you to understand the antibody mechanism of action much, much better. So let's talk a little bit about intellectual property. When you want to protect your antibody, there are really two key requirements, frankly, of any patent, and that's what is known as novelty and non-obviousness. In other words, is your antibody new? Is it unique? And is it unusual? Is it seeing something that's not obvious? Epitope mapping clearly is able to show antibody novelty by distinguishing novel mAbs from prior art mAbs. In the figure that's on the right, what you see are two antibodies that are binding to HER2. In green is a standard of care antibody, pertuzumab, with its prior art epitope in green. On the other hand, you can imagine a novel mAb, and this is some work that we did at Integral Molecular, where we epitope mapped a novel antibody, and it's clear that this novel antibody is binding a region that is distinct from the prior art epitope of pertuzumab. More interestingly, it appears that the ability to generate and to map conformational epitopes may contribute to the non-obvious antibodies. And there's some thinking that mAbs with conformational epitopes may be more patentable in part because mAbs with linear epitopes can be really considered obvious, since we routinely make these types of antibodies by peptide immunization. In addition, epitope mapping is a way of providing for disclosure of the antibody invention, which is one way that you show that you are in possession of the invention. And this is part of the written description requirements. We've previously had a detailed webinar on the role of epitope mapping in patents, and that's shown on the link below. So let's say you want an epitope map. What do? Well, there are a number of approaches for epitope mapping that are available. And when you're thinking about available approaches, what you want to ask yourself is, well, can the technique look at conformational epitopes for the reasons I just discussed? Is it high resolution? Does it get you down to real residue level epitopes? Can it do complex proteins? Can it do membrane proteins? Is it giving you energetic hotspots, the important residues that are contributing to the affinity of the antibody? And of course, speed is always important in a competitive research environment. And what I want to show you is that shotgun mutagenesis epitope mapping is able to fulfill a lot of these, these requirements for epitope mapping of complex membrane proteins. So let me tell you how shotgun mutagenesis epitope mapping works. The way we begin by epitope mapping is we engineer a parental plasmid expressing your protein of interest, the protein of interest, in living cells, and then we make a mutation library across the entire protein. This library can be relatively small. It can be hundreds of mutations. It can be thousands of mutations. We have a very efficient process, and we can generate these libraries in 2 to 4 weeks. We can then express each of these individual mutations individually in the well of a 384 well plate. And because of automation and of high throughput flow cytometry, we can analyze each individual mutation for the ability of an antibody to bind or to not bind. Those antibodies, those residues that don't bind, of course, those are going to be potential critical residues. Now, when you're epitope mapping these looking for antibody binding in these large libraries of mutations, you're, of course, generating a lot of data points by doing replicates, by looking at different antibodies. And we have proprietary algorithms and software that enable us to analyze these complex data sets. Most importantly is if you have a model or an actual structure of your target protein, we're able to actually integrate this binding data with structural analysis in real time to get a deeper understanding of these epitopes. And once again, because we're doing all this mutagenesis and expression in human cells, we can map conformational epitopes as well as linear epitopes, and we're looking at native protein structure, post-translational modifications. We're looking at the protein in the way you want to see it. So let me give you an example of validation of our approach towards epitope mapping. So what I have here is an example of a binding of an antibody to the Ebola envelope protein. So this is the part of the Ebola virus which is involved in getting the virus into the cells. So it's the key target for neutralizing antibodies. On the far left is a co-crystallography structure of the Ebola envelope glycoprotein with one of these antibodies. And in blue are the many residues that are present at the interface of the antibody protein of antigen binding interface. So that is the crystallographic epitope. We applied our shotgun mutagenesis mapping to the entire Ebola envelope protein, and what you see in the center panel are those residues that we pulled out by shotgun mutagenesis. Those are shown in green. As expected, only some of the residues that are defined by X-ray crystallography are contributing energetically. So these are the critical epitope residues shown in green. And in fact, they're where you expect these critical residues to be, which is generally at the core of the crystallographic epitope. One of the nice experiments that one can do with viruses is one can generate viruses that have resistance to neutralizing antibody, so-called neutralization escape residues. This has been done with Ebola with this particular antibody, and on the far right panel, in gold, you can see the residues that are mutated when you have a virus that is able to escape the neutralization by this antibody. And as expected, that residue that is mutated in the escape mutation, in the escape variant of Ebola, is within the critical residues that we define by shotgun mutagenesis. So this really shows you as validation of the shotgun mutagenesis approach and how it can really work, combine very nicely not only with crystallography, but also can predict where you may get escape mutations, for example, in viruses or, for example, in cancer. Here's another example where epitope mapping can really help you understand your antibodies better. This is another viral envelope protein. It's called chikungunya virus, and it's a very complex membrane protein. It forms a trimer. There's nice structural information available on this. We've been able to epitope map, in this study, 15 antibodies. We've epitope mapped many more since then. And in gold, we see those residues that are involved in neutralizing epitopes. And in green, you can see residues involved in non-neutralizing epitopes. We were able to isolate at integral molecular a unique antibody that neutralizes a chikungunya virus. That's shown in the box. It's known as IM-CKV063. And what we were able to show is that this antibody bound a very, very unique epitope on the chikungunya virus protein, indicating a unique mechanism of action. On the right, I show a detail of this antibody binding site. And unusually and unexpectedly, this antibody actually binds an epitope that's defined by the interface between two subunits. In other words, this is a quaternary epitope structure. So the epitope mapping was able to reveal how trimeric epitope was able to influence neutralization and was able to distinguish it from many other neutralizing antibodies. So if you're interested in a more detailed look at this antibody and other related antibodies, we have a nice publication by Fang et al. In the Journal of Virology, as well as a patent describing these studies in detail. Another interesting example, this time with the GPCR, is some work that we did with Novartis. And Novartis was interested in developing a biparatopic nanobody, essentially a bispecific antibody, which bound to CXCR2 at two different sites on the protein, as a way of generating a very potent antibody that would act as an antagonist for the function of the receptor. We mapped two of these nanobody units, and what we showed is that one of the antibodies, shown in the light beige circle, was a linear epitope. This was expected, while the second antibody, shown with the pinkish ovals, was clearly a conformational epitope, seeing residues on two different extracellular loops of the GPCR, as well as two different regions on one of the loops. And this is actually a rather unexpected epitope for a GPCR. And this work was also published in Molecular Pharmacology, ended up in a patent. And this epitope information was used to help decide which antibodies to use for the bispecific antibody. Our shotgun mutagenesis technique is a way of actually mapping large panels of antibodies. Over the years, we've done a lot of mapping work for biodefense pathogens, viruses that were of interest. And over a 4-year period, we were able to map really close to 400 different antibodies. That's shown in red there. And you can see that when we had the panels as we focused on each of the different antibodies, we were had a rapid rate of producing these different epitope maps for the different antibodies. And mapping these panels really can give you, enables you to decide which antibodies are the most interesting, which antibodies should move forward in any development process. Finally, we've been very interested in expanding the uses of these mutational arrays beyond epitope mapping. And I just want to briefly show you one example of this, where we were able to do functional mapping of a GPCR. This is a case of CXCR4. And what we did is we made a mutation array of the entire receptor, and instead of looking at binding, we looked at a functional assay. And what we were able to show is residues which disrupted the function of the receptor, and we're able to define residues which were involved in binding of the ligand, transduction of the signal through the membrane, and then show those residues that couple to the intracellular transduction machinery. And we've since expanded this work to now look at the binding of small molecule inhibitors to membrane proteins such as GPCRs. So really, this technology can be used for more than epitope mapping, although that is its primary function. If you're interested in this type of work, we just published a paper in PNAS several months ago, which can give you more details on this type of application of our shotgun mutagenesis platform. At this point, I want to change direction a little bit and just end this webinar on a discussion of another platform we have, our membrane proteome array, and in particular about improving drug safety. There was a recent paper in Nature Reviews which was looking about why small molecules drugs fail in clinical trials. And as you can see on the left, more so than efficacy or other types of failures, safety failures are the major reason why the projects fail during the drug development process. This is particularly true early on in preclinical and in phase 1, but is also a substantial reason for failures in phase 2. Why do molecules fail? In general, on the right, the reason they failed is not due to target-related adverse effects, target-related biology, but is due to the small molecule, the drug, binding and affecting off-target effects. Now, obviously, antibodies generally have fewer off-target effects than small molecules. But as we push the capabilities of antibodies with higher affinities, more complicated targets, more complicated functionalities such as bispecifics and multispecific antibodies, I think it becomes more and more important to look for off-target binding effects early on in the discovery process to minimize these potential safety liabilities. This is why we were interested in developing our membrane proteome array. What we've done in our membrane proteome array is we've taken 4,500 unique membrane proteins, and we're in process of expanding that to 6,000 unique membrane proteins, and we've arrayed them in 384-well plates and are expressing them on human cells. These are unique membrane proteins, not splice variants, so we're interested in looking across the entire membrane proteome of proteins expressed on human cells. We express these proteins on human cells or other mammalian cells to allow for native conformation, allow for native post-translational expression. We probe these expressed proteins using flow cytometry, which is a high sensitivity technique, and we look for the ability of the antibody to bind those bind or not bind an expressed protein. We have a number of applications that we're using this for. One is for specificity profiling, particularly to alleviate these safety concerns. We're also interested in deorphaning antibodies that may have been isolated by phenotypic screening, and also in general for new target discovery and for new biology. So let me give you a little bit more of a detail about how this works. As I said, we express each of these unique 4,500 membrane proteins in an individual well of a 384 well plate, and then run it through a flow cytometer and look for the ability of the antibody to bind or to not bind. This is exactly how we do the shotgun mutagenesis technology that I just discussed. As you can see on the left, you can see an example of an antibody where we've looked at the entire membrane protein library, and you can see that most of the membrane proteins do not show any binding of the antibody, while you see one particular protein, one particular target, showing a rather strong signal over all of the background binding. For those of you who regularly do flow cytometry and are more comfortable with histograms, if you look on the right, you can see an example just from a single 384 well plate. What you have here is 367 negative targets. So these are 367 distinct targets which do not bind the protein. This is shown in the gray, while the one hit on this plate clearly shows a shift at red. So it's a clear differentiation of a hit from background binding. Let me give you some examples. On the left, these are examples of antibody specificity profiling. For example, we started with looking at some FDA-approved drugs. This is rituximab. We profiled rituximab against the membrane protein library, and nicely, one target came up, CD20. That's the expected target. We've seen this with other FDA-approved drugs as well. We also decided, below that, we decided to look at some of our own lead candidates. This is one of our lead candidates for P2X3. That's the example that Ross Chambers showed. And what you see here is, of course, we have a strong signal against P2X3, but unexpectedly, we had an off-target binding to a unique protein. And what we've done now is this has enabled us to address this off-target binding early in the development process by affinity maturation and by looking at other potential leads. Another application on the right that we have for the membrane proteome array is convolution of orphan antibodies that may have been obtained from phenotypic screening, where you want to know what the target is. These are two examples where we isolated antibodies that see cells. We didn't know what they saw, though. So we ran it through our membrane proteome array, and these are two examples of orphan antibodies that we identified the targets for. On the top, one of the antibodies is binding SLC3A2, which is the subunit of a transporter, a cell surface transporter, and below we isolated an antibody against a tetraspanin protein, CD151. So both of these types of applications, antibody specificity profiling and deconvolution of orphan antibodies, really show how we can get a complete antibody specificity profile across the entire membrane proteome with very, very high sensitivity. I want to end with one final example. And of course, we're really interested in, can the membrane proteome array be used to screen for receptor function? And the answer is yes. And so, as mentioned previously, Integral Molecular has a strong, deep interest in virus and virus entry. And, given the recent outbreaks on Zika and Ebola, it was obvious that we could look for receptors that allow Zika and Ebola viruses to enter in the cells. We have technology at Integral Molecular that enable us to do make reporter viruses, where we can look for the ability of viruses to enter in the cells. What we did is we took the entire membrane proteome array. We expressed each of the individual members of the membrane proteome array in cells that were not permissive for either Zika or Ebola to enter, and we looked for the ability of the overexpressed protein to allow virus entry. And we were able to pull out receptors both against Zika virus and against Ebola virus. What I have shown here is just a portion of the membrane proteome array. We've since gone on to look at the entire membrane proteome array for these different viruses. We've pulled out a number of different receptors, some of which are known from the literature, and some of which are rather unique receptors. And so this has really added to the understanding of the deep biology of these different viruses. We're working on some manuscripts and really shows how the membrane proteome array can be used to screen for receptor function. Well, to summarize what Ross and I have talked about in this webinar, Integral Molecular, we have developed platforms that enable us to have a complete solutions to antibody discovery and characterization, soup to nuts, as it were. Ross showed you how by starting with a target, we can engineer the target so that it expresses well. We can present it on our lipoparticles, and then using a combination of DNA and lipoparticle immunization, get robust immune responses against difficult targets. Ross also showed you how we use both our microfluidic B-cell cloning capabilities as well as phage display to isolate antibodies with unique functions and diversity. We did not talk about our engineering capabilities, but we have some nice approaches to antibody humanization and affinity maturation, which I hope to be able to share with you at a future webinar. And then what I just discussed is antibody profiling, how you can use our membrane proteome array to understand exactly what targets does your antibody bind to allay safety concerns early on in the discovery process, and also talked about our epitope mapping, which can help you improve the intellectual property component of your antibody, as well as giving you deeper understanding of the mechanism of action of your antibody. And all of this put together really helps you generate good antibodies that are specific, have the function you want, are high affinity, humanized, and ready to go. For those of you who are interested in this technology and interested in working with Integral Molecular, we work with customers and partners in lots of different ways. Of course, we work with, we offer a number of our our technologies as reagents and surfaces. This includes our lipoparticle technology and shock immunogenesis epitope mapping, which many of you know us about. We also have our membrane proteome array, which is up and running, and of course, antibody discovery services. And Ross, of course, went into great detail in some of the work that we've done there. We also are always interested in working people on a co-discovery basis, either for looking for antibodies, potential therapeutic antibodies, or for novel target identification. And of course, within Integral, we've developed our own pipeline of potential therapeutic mAbs and engineered proteins, of course, all of our internal technologies, and we're always interested in licensing opportunities. At this point, I just want to show you, this shows a picture of the team at Integral Molecular, all of the great scientists who contribute to the science that we do. And on behalf of Ross and myself, I want to thank you for joining us for this webinar, and we'd be open to answering any questions at this point. Thank you very much.
Thank you very much, Dr. Rucker and Dr. Chambers. We have 2 pages of questions, so I don't think we're going to get to them all, but we'll give it a go. Our first question is for Ross. When screening for antibodies against the extracellular domain of a transmembrane protein, how do you design the epitopes so that they represent native conformations presented on the surfaces of cells?
Okay, thank you, Elizabeth. Yeah, so really our approach here is using DNA immunization, life particles, those platforms for delivery of the antigen to the animal delivers native epitopes. So it's really, we try not to engineer the antigen unnecessarily. We want to maintain those native epitopes. So by presenting the epitopes using DNA immunization and life particles, that maintains the native integrity of those epitopes.
All right. A second question for you. How do you estimate the scale of diversity required to ensure a high rate of true positives in screening process?
Yeah, so with the phase display screening, so we'll typically pan a couple of rounds and then start sampling there. So we'll screen a few hundred clones to see what the diversity is. And then to expand the diversity, we'll just keep sequencing and screening to expand that. And you'll typically get many different antibodies, different sequences by doing that.
Okay, perfect. And now a question for Joe. What types of proteins are in the proteome library? For example, 1 to 2, MTM.
We're, of course, interested in the entire proteome. So there's GPCRs, there's ion channels, transporters, various types of 1TM proteins, GPI-anchored proteins as well. So we really are going for diversity of the entire membrane proteome. And really, these are exactly the sorts of proteins that you'd be interested in, for example, as therapeutic targets.
All right. And another question. Can you determine the epitope for very high affinity mAbs?
Yeah, this is something we do that, and we do that on a routine basis. And I didn't go into detail in it in the webinar, but during the whole process of epitope mapping, we really do a lot of optimization of the binding, and we can look at antibody binding by looking at different stringencies. We can convert, for example, a bivalent mAb into a Fab, and by changing the stringencies, potentially going from a bivalent to a monovalent antibody, we can essentially tweak the affinity so that we get good epitopes. But we put a lot of effort into optimizing of the assay so we can pull out these critical residues. All
right. And a follow-up question, how confident are you in the hits you pull out from the proteome array?
Yes, we're very confident, and there's a number of reasons for that. One is that the way we do our assays with the flow cytometry on the living cells is our background binding is very, very low, and we can change cell types to optimize that. Because it's flow cytometry, our signals above the background are very, very strong, so we don't get false positives within the assay itself. Upon getting the hits, we confirm the hits in follow-up experiments, and of course, we sequence confirm any hits that we get. So we're very confident in the hits that we pull out.
Okay, and can you map soluble proteins?
Yes, absolutely. We can do that. Obviously, our own interest has been in membrane proteins, but one can certainly do that. For example, if you have a secreted protein, you can follow the exact same approach by mutagenizing the entire protein, and instead of looking at binding on the cells, you could look at binding onto the secreted protein. You could even do intracellular targets as well by doing a permeabilized flow cytometry, and we've done that before. Actually, we do that on a routine basis.
Okay, and what is your success rate for epitope mapping?
It's really high. I mean, it's probably greater than 95%. And just to go back to the earlier question, optimizing the binding assay by tweaking the stringency, by converting from a bivalent to a monovalent structure is really important. And so we go through that effort to ensure that we get an epitope out. So it's greater than 95%, significantly greater than 95%.
Okay, and this is for Ross. I'm going to take it back. We had several questions that came in on slide number 13, which I've just brought back up. The first question, is this example for a multi-pass membrane protein or a single pass?
Oh, both of these examples are for multi-pass membrane proteins.
Okay, and the second question on this slide, what serum dilution was used to determine the fluorescence shifts?
For many of them, it's 1 to 200, but we'll try a range, but typically 1 to 200.
Okay, perfect. Well, I see we're down to the very last minute, So what I will do is send along the rest of the questions that we've received to our presenters today. I would like to very much thank both presenters, Dr. Ross Chambers and Dr. Joseph Rucker. I'd like to thank Integral Molecular for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent some time with us. We appreciate you coming, and we hope we were able to provide some answers to your research questions. So with that, I'll say thank you all so much, and have a great day.